G proteins are the first type of receptor molecule. The ligand will initiate the communication by binding to the G protein link receptors in the cell membrane. The G protein itself is currently inactive and has GDP, shown in light green. It then moves toward the receptor protein, which phosphorylates the G protein. The G protein now contains GTP and travels to activate an inactive enzyme, which then changes form and starts a cascade of energy transfer until the cell response is elicited. In this case, the goal of the pathway is to penetrate the nucleus, activate a specific gene, which commands the cell to produce more of a specific protein, hence the ribosomes that come out of the nucleus.
This is the Tyrosine-Kinase Receptor system. As a pair of ligands bond with the receptor kinases, the kinases move together forming a dimer. ATP will then phosphorylate the tyrosines attached to the kinase. A signaling protein then receives the signal from the tyrosine and may send it to an enzyme such as adenylyl cyclase to produce a second messenger like CAMP. The signal is then transduced throughout the cell and may be amplified. In this example, the cell responds by sending the signal to the rough ER where calcium ions are released.
Although the cause of multiple sclerosis is unknown, a common theory suggests that an unknown factor mysteriously causes the neuron to seem like a pathogen. This probably works with the association of the neuron's antigens as being foreign. As soon as the T cell's antigen receptors are activated, they reproduce and tag the neuron with cytosines. This later brings in the macrophage to destroy it. As soon as the macrophage is done, this results in major damage to the myelin sheath.
During synaptic signaling, the synapse allows electrical energy of the presynaptic cell to convert a chemical energy for the postsynaptic cell. When action potential reaches near the end of the presynaptic cell, it activates voltage-gated Ca2 channels, allowing calcium ions to flow into the terminal. The increase of calcium concentration caused the release of neurotransmitters. The neurotransmitters diffuse across the synapse and bind to receptors that open ion-gated channels. The flow of ions depolarize the postsynaptic cell and convert the neurotransmitter's chemical signal into an electrical action potential. When the myelin sheath is destroyed during multiple sclerosis, the axon is unprotected and exposed, which causes nerve signals traveling down the axon to be slowed or even blocked. This slowing down and blockage of the nerve signals cause the symptoms associated with multiple sclerosis.